Hello class, today we're going to be covering chapter 15, which is metabolism and nutrition. I've broken this lecture into 3 different parts, so this is the beginning of part 1. So we just finished digestion and we're going to be moving into metabolism. The products of digestion that reach our body cells are monosaccharides, fatty acids, glycerol, monoglycerides, and amino acids. Now, what happens to this absorbed food molecules? Most of it is used as energy for our life processes. Some of it is used as building blocks for more complex molecules, and some of it is stored for future use, like glycogen is stored in liver and triglycerides in adipose tissue. Metabolic reactions harvest the chemical energy in these products of digestion. So to begin with, metabolism refers to all the chemical reactions taking place in the body. Reactions that break down complex molecules into simpler ones are called catabolic or catabolism. These are decomposition reactions. Typically, these are exergonic reactions, which means they produce more energy than they consume, releasing the chemical energy. For example, glycolysis, the Krebs cycle, and the electron transport chain are examples of catabolic reactions. Reactions that combine simpler molecules to make a more complex one are called anabolic reactions. These undergo a synthesis reaction, so you're synthesizing something new. They tend to be endergonic, meaning they consume energy more than they produce it. So they need energy input for these to occur. Examples of this include building peptide bonds in protein synthesis or linking glucose molecules in order to form glycogen. If you could please pause this video and watch Introduction to Metabolism now, and then come on back. So metabolism results from the balance of anabolic and catabolic reactions. ATP, or adenosine triphosphate, is the energy molecule that couples the two of these types of reactions. It's considered our energy currency. So our body creates ATP and then uses it to do things in the cells that require ATP. With ATP, a phosphate group breaks off of it, releasing energy. So ATP is adenosine triphosphate, so it's an adenine molecule with 3 phosphates. When we break one off, we release energy, and it becomes ADP, adenosine diphosphate. Okay. The release of energy is used to fuel the anabolic or endergonic reactions, requiring energy to build new molecules. Here we see the linkage between the anabolic and the catabolic reactions. We see in the middle, the thing that is linking them both is ATP being hydrolyzed into ADP and PI, and that energy is then going to fuel the reactions that require energy. Also, I want you to notice that heat is released at different parts of this reaction cycle. So remember, metabolism is all of our reactions in our body, and they would include both the catabolic ones, which are breaking down molecules, and the anabolic ones, which are building more complex ones. So energy transfer. Molecules have stored potential energy in their bonds. Oxidation reduction reactions are one category of reactions important in the energy transfer. You may have heard of this in chemistry. So an oxidation reaction, involves the removal of electrons from one atom or a molecule. The removal of electrons decreases the potential energy of that molecule. An example of this conversion an example of this is the conversion of lactic acid to pyruvic acid. So this oxidation, we're removing electrons. So here we have lactic acid, and in the oxidation of it, we remove 2 hydrogen ions, an H plus and an H minus, okay? So when we do that, we're moving one here and moving this one here. We end up with this compound called pyruvic acid. The opposite reaction of oxidation is reduction. This involves adding electrons to a molecule. The addition of electrons increases the potential energy of the molecule because you're adding bonds. An example of this is converting pyruvic acid to lactic acid, so the opposite. So here we have pyruvic acid, and then we add 2 hydrogen ions to create lactic acid, and this is a reduction. So over here I put, isn't it weird that a reduction reaction adds stuff to it? But that's correct. That's how it works. We add electrons, we add potential energy to one molecule to create something new. So when a substance is oxidized, the liberated hydrogen atoms are immediately transferred by 2 coenzymes to another compound. So they don't just hang out in the atmosphere. The 2 coenzymes are nicotinamide adenine dinucleotide, NAD, this is how we know it as, and flavin adenine dinucleotide, or FAD. So we have these 2 coenzymes that are hanging out. They will take up the 2 hydrogens and then result in NADH and a hydrogen. Or it can be, and that would be a reduction, okay? Or we can break apart that bond and have the oxidation. The same thing goes for the coenzyme FAD, okay? It can become reduced in one direction and it can become oxidized in the other. So these are coenzymes, they're kind of the intermediaries. Oxidation and reduction are always coupled together. So the 2 reactions together are called oxidation reduction or redox reactions. The oxidation of lactic acid to pyruvic and the associated reduction of Na plus may be written as such. So when we want to reduce lactic acid to pyruvic acid, when we want to oxidize lactic acid to pyruvic acid, then we will reduce NADH into NADH plus pH. So in each of, as this happens, the other one happens. As an oxidation happens, a reduction happens. So it's a redox reaction. All right, so how does this all relate to ATP and the food we just ate and digested? So some of the energy released during oxidation reactions is captured when ATP is formed, okay? So a phosphate group can be added to the ADP in a process called phosphorylation, along with energy to form ATP. So the adding of that phosphate group creates a bond, and in that bond is this energy, okay? So adenosine with 2 phosphates is ADP. We add a phosphate to it, and we have to add energy into it. That allows a new bond to be formed, and we now have ATP. So phosphorylation mechanisms to generate ATP. Remember, we want ATP so we can do everything else in our bodies, right? So we need to make all this ATP as much as we can, and our body does that through a bunch of different ways, but it's basically we're phosphorylating ADP. Okay, that's how we make it. We got ADP hanging around, we put the phosphate back on it, now we got ATP and our cells can use it to move stuff, build things, whatever requires energy. So 3 ways to phosphorylate ADP into ATP. Substrate level phosphorylation. This is the transfer of the phosphate group from an intermediate compound or a substrate directly onto ADP. This occurs in the cytosol of our cells, so in the cytoplasm. There's also oxidative phosphorylation. This removes organic compounds and passes them through a bunch of electron acceptors called the electron transport chain to molecules of oxygen. This occurs in the inner membrane of mitochondria. And then the last one is photophosphorylation, and we don't do this. This only occurs in plants and some bacteria, anything that has chlorophyll. So this one doesn't happen in our body. However, substrate level phosphorylation and oxidative phosphorylation does. So let's start with carbohydrate metabolism. This is our big topic for the first video. So in carbohydrates, the digestion of carbohydrates hydrolyzes them into, the carbohydrates are polysaccharides and disaccharides, and we hydrolyze them into monosaccharides. So we got this final compounds of monosaccharides. 80% of those monosaccharides are glucose, but we also have some fructose and galactose. All three of them are monosaccharides. And the fructose and galactose gets converted to glucose by our liver. So carbohydrate metabolism is mostly glucose metabolism. The body's use of glucose depends on the needs of the cells. Our cells may need ATP production, amino acid synthesis, glycogen synthesis, or triglyceride synthesis. Okay, so that's what glucose can do for us. So ATP production. Glucose is oxidized to produce ATP for immediate energy. In amino acid synthesis, we use glucose to form amino acids for our proteins. Glycogen synthesis is also known as glycogenesis. So it has the word glycogen in it, and then genesis means the building of something new. So glycogenesis. So to make glycogen, our hepatocytes in our liver, those are just liver cells, and muscle fibers combine hundreds of glucoses to form glycogen. And we use glycogen for storing this energy. And then lastly, triglyceride synthesis. When the liver's glycogen storage areas are full, and we still have more glucose, the hepatocytes transform glucose into glycerol and fatty acids. Transform glucose to glycerol and fatty acids for use in lipogenesis, which is the synthesis of lipids or the synthesis of triglycerides. So excess sugar, excess glucose is going to be converted to fats, triglycerides that are stored in our fat. That's why when we eat too much junk, we've already stored enough glycogen and we have excess glucose is converted to fat. So please pause this video now and go ahead and watch carbohydrate metabolism. This is a good one you may want to watch later so that you can, as part of your studying, because carbohydrates are by far the most complicated of the nutrients that we have to metabolize. And then come back. All right, hopefully that video didn't complicate things too much for you because it did introduce some topics we're going to discuss now. Glucose must pass through the plasma membrane in order to be used by the cell. Facilitated diffusion makes this happen because glucose cannot just go straight through. And most cells, the GLU-T molecules perform this. The GLU with a capital T. Insulin increases the insertion of GLUT4 transporters into the plasma membrane of the cell, increasing the rate of facilitated diffusion. Okay, so GLUT molecules are like little transporters. They're in the cell membrane, and they're allowing glucose to go through. Insulin makes more of these GLUT molecules go into the plasma membrane. So that makes more sugar go into the cells. More glucose goes in the cells because insulin, remember, decreases blood sugar. So when we have too much glucose in our blood, we got to put it into cells. So inside the cell, the glucose is phosphorylated. So we're adding a phosphate group. GLUT cannot transport phosphorylated glucose back out. So now it's trapped inside the cell. Okay. So this brings us to cellular respiration. Oops. Cellular respiration is the oxidation of glucose to produce ADP. Okay, remember that's going to be our first big use of glucose. So oxidating glucose produces ATP, water, and carbon dioxide. So this is why, because of some of the processes in here, this is how we produce carbon dioxide to exhale. We're going to use oxygen, which is why we need to inhale oxygen, make some ATP, and then we're going to exhale this product, carbon dioxide. Remember, oxidation reactions are usually exergonic, releasing energy, which then can be used to phosphorylate ADP to ATP. Okay? This occurs in the cells with our mitochondria. So the only cells we have that don't have mitochondria are red blood cells. So this does not occur in red blood cells. So there are 4 sets of reactions involved in carb metabolism or cellular respiration. The first is glycolysis. The second is the formation of acetyl coenzyme A, then the Krebs cycle reactions, and lastly, the electron transport chain reactions. So these go in order as to how the cell would use them. Glycolysis is the only set of reactions that don't require oxygen. So they are considered anaerobic reactions. Aerobic reactions require oxygen, which are the number 2, 3, and 4. Now, glycolysis, in order for it to occur, it has to use 2 ATP. But through its process, it produces 4 ATP. So really the overall net production, the net overall is we make 2 ATP from glycolysis. These reactions take 1 glucose and do a bunch of stuff to it. And then it makes 2 pyruvic acids, 2 NADH plus H plus, and 2 ATP. Glucose has 6 carbons. So when we break that apart, we're going to get 2 3-carbon molecules, and those are our pyruvic acids, as well as this other stuff. Okay. So if we don't have any energy available, this is all that's going to happen. I mean, any oxygen available, this is all that's going to happen. We're going to end up and be able to make 2 ATP. It takes a long time. It's slow. We're only getting a few of them. And we're going to go through our glucose pretty quick. The second way, the formation of acetyl-CoA requiring oxygen. This is the transition step between glycolysis and Krebs. In this step, we take the 2 pyruvic acids that we made in glycolysis and convert them into 2 acetyl coenzyme A's. Remember, we just talked about how we can convert stuff like that. As well as we get 2 carbon dioxides and 2 NADH plus H plus. So again, we're making some more of these intermediaries. Now, the Krebs cycle and electron transport chain, this takes the 2 acetyl coenzyme A's and breaks it down into 4 carbon dioxides, 6 NADHs and H pluses, 2 ATPs, and 2 FADH2s. Okay, so this is another one of the, I showed it in the previous slide, another one of these intermediaries. Then lastly, the electron transport chain takes all of these intermediaries and some hydrogens, because that's also part of this here and here with the NADH. They get run through the electron transport chain, and we end up getting 28 or 30 ATP. Okay, it kind of depends how certain things work, but that's the number 23 or 30. 6 molecules of oxygen and 6 waters. Okay. All right, so here we see carbohydrate metabolism with all the steps. Glycolysis is taking 1 glucose and making 2 pyruvic acids and 2 ATP, 2 NADHs plus 2 H plus. Two pyruvic acids enter the formation of the acetyl-CoA. That's going to be converted to two acetyl coenzyme A's and two CO2s plus two NADH plus two H's. The two acetyl-CoAs enters the Krebs cycle. The Krebs cycle creates, after its several processes, two ATP, four CO2s, six NADH plus six H pluses, and two FADH2s. All of these blue boxes go into the electron transport chain, bing, 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 and off of it we get 26 or 28 ATPs, 6 oxygens, and 6 waters. Now, the total amount of ATP that can be made from one glucose molecule, if in the presence of oxygen, is going to be this 2 plus these off of the electron transport chain, plus these off of the Krebs cycle. So we got 4 here, add it to the 26 or 28, and we have 30 or 32 ATPs. Getting a little more personal with the different steps. Glycolysis is a process where a 6-carbon glucose molecule is split into 2 3-carbon molecules of pyruvic acid. It does not require oxygen, therefore it's considered anaerobic. It uses 2 ATP, but produces 4, so the net overall is a production of 2 ATP. Here is the reaction. 1 glucose creates 2 pyruvic acids, 2 NADH plus H pluses, and 2 ATP. Glycolysis involves 10 reactions that you do not need to know. So glycolysis is right here. We see how it's broken into 1 of these, mixed 2 pyruvic acids, plus the ATP, NADH, and H plus. This is the overview of glycolysis. We have this 6-carbon glucose. We have to use 2 ATPs, and then we create boom, 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 2 3-carbon pyruvic acids. We then make 4 ATP, 2 here and 2 here, and 2 NADH pluses plus H plus. Okay. So what happens to the pyruvic acid? Depends on if there is oxygen available. If there is not oxygen, there are anaerobic conditions, the pyruvic acid is reduced by adding 2 hydrogen atoms and forms lactic acid. If oxygen is plentiful, like in aerobic conditions, most of those cells are converted to pyruvic acid, from pyruvic acid to acetyl coenzyme A. So here we have the fate of pyruvic acid. There's the glucose. We create 2 pyruvic acids. If there is no oxygen, then boom, 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 2 lactic acids. If there is oxygen, pyruvic acid in the cytosol gets moved into the mitochondria, in which it goes and creates the 2nd step, formation of acetyl coenzyme A. Okay, so here's the reaction for the 2nd step. 2 pyruvic acids get converted to acetyl-CoA plus 2 carbon dioxides plus 2 NADH plus H plus. All right, 3rd step, the Krebs cycle. Dum, dum, dum. So we've done glycolysis, we got acetyl-CoA. The acetyl-CoAs are going to enter this Krebs cycle. This Krebs cycle is very complicated. This is the simplified version of the Krebs cycle. There's a more in-depth one as well found in your book. It goes into all the different conversions. So basically we have 2 pyruvic acids coming through, so everything here is doubled for 1 glucose. So then we end up, per pyruvic acid, we get a molecule of CO2, NADH plus, or NADH plus H plus, converted into acetyl-CoA. Acetyl-CoA goes into this citric acid cycle and creates NADHs here, which are going to go to the electron transfer chain, some ATP, and then FADH2 and NADH again. So all of these blue boxes are heading to the electron transport chain. And you see it down here. The Krebs cycle is also known as the citric acid cycle. Two pyruvic acids enter the matrix of the mitochondria and undergo 8 reactions of the Krebs cycle. They are oxidation reduction reactions, and decarboxylation reactions release the CO2. Overall, the net reaction of the Krebs cycle is 2 acetyl coenzyme A's get converted to 4 CO2s plus 6 NADH plus H plus 2 ATP and 2 FADH2s. All right, so all of those NADHs and FADH2s get sent to the electron transport chain, which is a series of electron carriers in the mitochondria. Each carrier in the chain is reduced as it picks up electrons and then oxidized as it gives up the electrons. Exergonic reactions release energy, and that is used to form the ATP. This mechanism links chemical reactions with the pumping of hydrogen ions and is known as chemoosmosis. Okay. So the overall reaction is 10 NADHs plus 10 H pluses plus 2 FADH2s. They all enter the electron transport chain, resulting in 28 or 30 ATPs, 6 oxygen, and 6 waters. Shows the electron transport chain. It's pretty complicated, but it's also very cool in the way that it is structured. So on the bottom, I've also just included the reaction again for the electron transport chain. So in here, we have the mitochondrial membrane. This is inside the mitochondria. This is outside the mitochondria, okay? It's still in the mitochondria, but it's the inner membrane. So it's like in the inner part. And then this is the kind of the outer part, okay? You see how that is? So we have all in the inside, we have NADH and H pluses that got delivered here. They go through this electron transport chain, and when they do, H plus is created here. We have a high H plus concentration between these two membranes. They then go through this channel, and this is the H plus channel, and this whole thing is ATP synthase. So as the hydrogen, it's going to want to go from this side to this side because of the concentration gradient. So as it comes in here, because this is happening by the gradient, goes through there, and then that energy is going to be harnessed into making ATP from ADP plus P, okay? So this keeps going. Some of the electron carriers include flavin mononucleotide, FMN, cytochromes, iron, sulfur centers, copper atoms, and coenzyme Q. Cellular respiration, which is all of those reactions, those 4 stages that we talked about, will generate either 30 or 32 ATP molecules for each glucose molecule that is catabolized. The overall reaction of glucose oxidation is, this is taking all 4 steps, this giant reaction. We have glucose, we add some oxygen, 6 molecules, and 30 or 32 ADPs, and 30 or 32 phosphates. It goes through all those reactions. Overall, 6 carbon dioxides are made, 6 waters, and 30 or 32 ATPs. So this is the location of these different reactions. We have glycolysis happening in the cytosol. We have everything else happening in the mitochondria. So that was one thing that glucose can give us. We can use it to make ATP. If we don't need it immediately, it can be stored as glycogen. The process converting glucose to glycogen is called glycogenesis. It happens in the liver and skeletal muscles and is stimulated by insulin. When ATP is needed for our body activities, the stored glycogen can then be brought back and broken down by a process called glycogenolysis. The lysis means breaking down. So I say it in order to emphasize that part, glycogenolysis. I think it's I'm gonna say it wrong now, glycogenolysis, but I like to hear lyse because lyse means breaking apart. Now this process is stimulated by glucagon and epinephrine, so those hormones, and this is a hormone as well. So if our blood glucose is too high and we need to do something with it, we then bring it inside this liver cell. It can be converted through into glycogen. So this process is called glycogenesis. We store it here a whole bunch, and then all of a sudden, oh, and also this movement, the glycogenesis, is stimulated by insulin. So insulin is excreted when we have too much glucose in our blood. Then we have glycogen hanging out in our liver cell, and all of a sudden we need some more energy. We haven't eaten in a while, and we need our blood sugar to go back up. So the reverse happens. We convert it to glucose, and then glucose gets released into the blood. And this process, the blue arrows, is called glycogenolysis, and is stimulated by by glucagon and epinephrine. I want to point out here this intermediary, because we're going to discuss this later. The intermediary of this glucose transformation to glycogen is glucose 6-phosphate. So just pointing that out, it's for later. Glucose can also be formed into proteins, as well as the glycerol portion of triglycerides, lactic acid, and certain amino acids. This process is called gluconeogenesis. Glucagon, cortisol, and thyroid hormones stimulate gluconeogenesis. All right, so gluconeogenesis is stimulated by cortisol and glucagon. They take lactic acid, different amino acids, some glycerol, and they are putting it into this reaction. So we have some pyruvic acid sitting around. They can take these guys and create more glucose. I want to point out as well the glucose 6-phosphate here. So gluconeogenesis. If you look at that word, it has glucose in it, and then new genesis is making something new. So it's almost like taking something that doesn't have anything to do with glucose and converting it into a new glucose, okay? And there again, just look, there's our glucose 6-phosphate. And this is our end of our first video. I will see you in part 2.